Hello, Jeff Hansen here again. This week we talked about amino acids and proteins. It turns out that a very important type of protein is an enzyme, and enzymes often work in concert with coenzymes. So today I'm going to give a little bit of an introduction to enzymes and to coenzymes. Some of this will overlap what we've talked about before, but I felt that some people had difficulty in understanding the nucleophilic attack and what's behind ATP. So today I'm going to go a little bit deeper and into more detail. So even if you understand ATP, I think it's in your interest to work through this anyways and see if you can get even a stronger or deeper understanding of it. And this will set the context then for looking into a number of different coenzymes. For the learning objectives, I want you to explain a little bit about catalytic mechanisms. I want you to explain in detail why it is that ATP has high energy bonds and what the role of those bonds are in making life work. Then I want, in part 2, to look at each of the B vitamins and get some idea of their roles, and I want you to zoom in, especially on NADH and FADH, in order to explain how they play such central roles in metabolism. Enzymes are proteins that catalyze chemical reactions. They depend upon many different mechanisms. They might involve proximity and orientation, proton donors, proton acceptors, electrostatic, covalent, metal ion. But among all of these mechanisms, one is absolutely essential, so that regardless of the catalytic mechanism, all enzymes will include at least one step that requires what catalytic mechanism. I'd like you to pause the video and take a quick stab at that, but I'm going to move on. Fundamental to any reaction is proximity and orientation. If you think about it, this is an obvious answer. Why? Well, how can you form a bond between two substrates unless those substrates are close enough together to form a bond in the first place? Furthermore, given that the two substrates have been moved close enough to form a bond, in order to form a specific bond, you will have to have the correct part of one substrate near the correct part of the other one. In fact, close enough to form a bond again. It's only after you've moved things close enough together and pointed them together in the right way that now other catalytic mechanisms can contribute, such as acid-base catalysis, metal ions, charge stabilization. Right now, I hope that each of you is following this very well and you're thinking, okay, this is so obvious, I don't have to rely on memorization at all. The point makes sense. But to be sure, because this is so important, I'm going to ask one more question. If substrate 1 and substrate 2 are not close enough together to form a bond, is it possible that any other mechanism could speed up the rate at which they react, such as charge stabilization, acid bases, metal ion binding? And the answer is no, they've got to be close enough first. So all mechanisms first rely on positioning, orientation, and then other mechanisms can contribute and speed up the rate even more. If that doesn't make sense, don't hesitate to email me or phone me. Now, there's many different types of substrates that can be bound by an enzyme in order to react. Number one, you bind the substrate. Commonly, enzymes will bind two substrates, one of which might be a simple water molecule. The enzyme positions the two substrates right next to each other and orients them to favor a chemical reaction. 90% of the chemical reactions that are catalyzed in your body incorporate a nucleophilic attack step. Essentially what that means is that the two substrates will be placed very close together, One of them will point a pair of electrons exactly toward an electrophile, probably a carbon atom on the adjacent one, so that the two are so close together that the chemical reaction occurs, you form the product, and it comes off the enzyme. Whereas many of life's reactions involve two distinct substrates, often one of the substrates is actually a coenzyme, such that the coenzyme can bind, the reaction occurs, and the coenzyme might even donate a chemical group to the substrate to make a product. Then the coenzyme is regenerated so it can cycle through the reaction. I think that everyone listening to this video is aware of a very prevalent and important coenzyme. It's so prevalent that it's often called the currency of intracellular energy. It's in your interest to go ahead and answer this question. I think most of you know the answer. How about ATP, adenosine triphosphate? Do you think ATP is an important molecule? For example, how many times do you think you've heard the word ATP used in your biology courses? I'm predicting it's over 1,000 times, and you've seen over and over and over that it is the central currency of energy for the cell. Accordingly, ATP is the molecule which links the calories in our diet with what powers our brain, our muscle, our immune system, our growth, our life. And it's at the center of a lot of diseases. I want you to be able to explain in detail why the phosphate bonds in ATP are high in energy. And to ensure that, I will be testing on this concept too. So please take your time and do not memorize what's coming up. Instead, ask yourself, do I understand the nature of the high energy phosphate bonds in ATP? And do I understand mechanistically how that energy is used to power life's reactions? In order to understand the function of ATP, first we must understand its structure. What I think is remarkable about ATP is the different types of groups it has. First of all, there's an adenine base. This is a largely aromatic and greasy part of the molecule. Then it has a ribose sugar, which can hydrogen bond. And then it has some charged groups. So you go from hydrophobic to hydrophilic to extremely hydrophilic. This is perfect if you want a compound that can be bound and oriented by an enzyme. For example, in the binding pocket of an enzyme, you might have represented in brown a very greasy part that will bind that adenine and hold it in position. And you might have some hydrogen bonding groups that will then interact with the ribose, and you often will have some positive charges that will interact with the negatively charged phosphates. Now notice the nomenclature alpha, beta, gamma, simply saying the phosphate closest to the ribose, phosphate next closest to the ribose, phosphate next closest to the ribose. All three of these phosphate bonds would love to be broken. Can you tell me why? Please pause and answer. If you do pause and can figure it out, you're in very good shape. These bonds would love to be broken because like charges repel. This large amount of negative charge does not want to be so constrained into such a small region of space. We've all seen the Van Graaf generator demonstrations. If you add a bunch of static charge to hair, the hair stands on end and repels and pushes it apart. Now consider 4 negative charges held very tightly together in a few angstroms distance by these covalent bonds. If we can break any of the bridging oxygen bonds, then some of those charges can be allowed to separate. In fact, here I'm showing hydrolysis. A water molecule nucleophilically attacks the partially positively charged phosphorus atom. The electrons resonate up on the oxygen, resonate down, and kick off adenosine diphosphate, and the inorganic phosphate comes off. At this point, can you predict what direction this inorganic phosphate will move spontaneously? It's not going to move toward those negative charges. Like charges repel. That inorganic phosphate is going to fly away. this is going down in energy. Anytime you break a phosphate bond, you're going down in energy, the charges separate. And a very important job in the cell is to take the negatively charged phosphate and the negatively charged diphosphate on ADP and to push them back together, and it takes energy to make ATP. That's why you burn sugar and fats. In fact, this reaction, hydrolysis of ATP, is so prevalent that if I make a solution of ATP in my lab, I have to freeze it at -80 to keep it stable. If it sits out on the counter for an hour or for a day, it's pretty much wasted. And I'm like, well, wait a minute. How do we maintain such a high concentration of ATP inside our cells? It is true that ATP in your cells right now is being randomly hydrolyzed. But at the same time, you're making massive quantities and using it before too much of it gets hydrolyzed. Life is expensive. Not only does hydrolysis of a phosphate group on ATP allow the charges to separate, allow that repelling charges to move apart and go down in energy, but in addition, it allows an increased resonance. So there's two ways the charges can spread out in space. Most of the organic figures that I'll use throughout my talks do not specifically label carbon atoms nor hydrogen atoms that are linked to carbon. Rather, they're represented as bends in lines or intersections of lines or as the termination of a line. However, I do label all of the oxygen atoms, phosphorus atoms, nitrogen atoms, sulfur atoms not shown, or hydrogen atoms that are linked to an oxygen or a nitrogen or sulfur. Also, I usually don't show the lone pairs of electrons. To be sure we're all on the same page here, will you identify all of the atoms that should have lone pairs of electrons here and tell me how many lone pairs each of those atoms should have? And if you're having any trouble at any point, I do want you to call me or email me. I do think the majority of you will find this easy, and some of you will even be bored by it. But if you're having trouble, I think it would be a mistake to move on when you could resolve this by talking with me for just a few minutes. I do expect that most of you will periodically jump over to Wikipedia for a few minutes to clarify the terminology. However, I certainly do not want anyone to spend hours in such searches or agonizing in trying to figure out what I'm saying. Maybe staying up half the night, banging their head against the wall. This is not meant to be complicated. In fact, sometimes what I say is so simple that you're thinking, well, I must be missing something. No, a lot of it is very simple and I'm repeating myself, but I want to be clear. So if you get stuck, give me a call. If you're getting a headache, give me a call. But let's move on. Well, from the octet rule, we know that nitrogen, carbon, and oxygen each are going to be surrounded by 4 pairs of electrons, either in bonds or in lone pairs. Whereas in contrast, phosphorus is one row below oxygen and carbon and nitrogen in the periodic table, so it will have larger underlying clouds of electrons, which mean in the outer shell there will be room for 5 pairs of electrons. So here it'll have 3 pairs of electrons in single bonds to oxygen and 2 more in a double bond to oxygen. So if an oxygen has 2 bonds to it, in addition, there will be 2 lone pairs of electrons, which aren't shown here. If an oxygen atom has only 1 bond to it, then there will have to be 3 lone pairs of electrons. If a nitrogen atom has 3 bonds to it, then there will have to be 1 lone pair of electron. That tends to complicate these figures, but sometimes I need to show those lone pairs of electrons, especially when I'm going to be talking about resonance or repulsion between lone pairs of electrons. And of course, the reason that phosphate groups tend to be acids is the same as why carboxylic acids tend to be acids. If the positively charged protons come off and leave a negatively charged lone pair of electrons, those negatively charged lone pairs can then resonate among all of the available oxygen atoms. For example, here I'm showing a negative charge on each of the lower oxygen atoms. That lone pair of electrons can resonate up to form a double bond. Meanwhile, The double bond to the upper oxygens can resonate up on top of them so that now the negative charge is up top. I chose to draw the resonance around each phosphorus atom in parallel simply so I didn't have to make a dozen different figures of every permutation. To be clear, the resonance around each of the phosphate group is pretty much independent of the other phosphate groups. And in fact, the way to think about it is that we have a cloud of electrons around each phosphorus atom independently resonating there. This figure also emphasizes that it is the branching oxygen atoms around which the electrons are resonating and not so much to the bridging oxygen atoms. Why isn't there much resonance to the bridging oxygen atoms? Certainly it is possible that a lone pair of electrons on the bridging oxygen can resonate down to form a double bond with a phosphorus, such that it would be an oxyanion above and an oxyanion below the phosphorus. The problem with this configuration is that the extremely electronegative oxygen is going to have a formal positive charge because what was a lone pair of electrons belonging to the oxygen is now in a bond to the phosphorus that it has essentially lost the equivalent of one of its negative charges, and oxygen is so profoundly electronegative that this happens very little. Of course, when a water molecule nucleophilically attacks the gamma phosphate and that gamma phosphate comes off of the ATP, that will eliminate one of the bridging oxygens and thereby will free up more resonance to again let negative charge spread out more and go further down in energy. Please pause the video to show the nucleophilic attack of a water molecule on the gamma phosphate of ATP. You should only understand one organic mechanism because in that way you will understand 90% of the chemistry going on in your body right now. So one mechanism is what I teach. It'll be in every talk. If you can gain that understanding in this talk and the next couple, you'll be in good shape. So let me show that. A lone pair of electrons on water, negatively charged, will nucleophilically attack the phosphorus atom. That is also partially positively charged because look, it's linked to 4 oxygen atoms, all of which would like to pull electrons away from the phosphorus. So the water wants to attack it, negative charge interacting with a positive charge. As the lone pair of electrons begin to form a bond to the phosphorus atom, the electrons in the double bond between phosphorus and oxygen will resonate up onto the oxygen to form an oxyanion. I do expect I will have tests where you have to identify an oxyanion. Can you interpret it? It sounds simple. Oxy, it's an oxygen atom. Anion, it's negatively charged. It only has a single bond to some compound, which leaves a negative charge on it. Now, I have to point out that this oxyanion is no different from this one or this one. My point is that a nucleophilic attack on a substrate will almost always generate an oxyanion in the intermediate, and then it is resonance of that lone pair of electrons on the oxyanion that can resonate back down and reform a double bond, resulting in pushing away a pair of electrons onto the leaving group. We want to break inorganic phosphate off of the triphosphate. Can you draw arrows to show how electrons will resonate to break the bond and have inorganic phosphate come off? Please pause and take a stab at it. Again, if you haven't had organic recently or feel a little intimidated, you're going to get more of this in the future. Take a quick stab, but I'm going to move on. Okay, we could draw the electrons resonating from any one of these oxyanions to reform a double bond, much like we have here. And as it does that, we're going to want to break this phosphate away from this phosphate. Therefore, the electrons that link the phosphorus atom to the oxygen will resonate up onto the oxygen, and that will give us an oxyanion, an ADP, and as the electrons resonate down here, we will have inorganic phosphate come off. Now, what will happen? The second these negative charges separate, down in energy. That's why ATP bonds are high in energy. In fact, these will tend to move apart rapidly, and to rebuild ATP is going to take a lot of energy to force them back together. Now I want to repeat. The reason ATP is so high in energy is when you break it, charges separate. But it's not just that the negative charges on the diphosphate repel the negative charges on the inorganic phosphate, but the very fact of breaking the bond changes the nature of the phosphate group that comes off. Inorganic phosphate is much more symmetrical than are the phosphate groups bound to ATP. Therefore, without that bridging oxygen atom, the electrons can resonate freely among all four oxygen atoms on inorganic phosphate equally, which means the electron cloud can spread out in space, and that's exactly what charges want. A light charge wants to spread out. This therefore goes down in energy due to resonance. So we've looked at nucleophilic attacks, we've looked at ATP. Now I want to show how ATP can use its energy, at least one way it can do so. And let's look at an enzyme. Hexokinase. Can you interpret it? All these names sound like Greek, and a lot of them are. Hexo means 6. O is short for os, which means sugar. Kinase means to add a phosphate group to a substrate. So this is hexokinase is an enzyme that adds a phosphate to a 6-carbon sugar. In particular, hexokinase grabs glucose and ATP. It positions them together where one of the hydroxyls of glucose can nucleophilically attack the ATP, much like in the reaction we just saw with water. Here I'm showing ATP again, here I'm showing glucose. And we will talk a lot about glucose. I will simply say that this is carbon 6 here and oxygen 6, and hexokinases catalyzes the transfer of the gamma phosphate of ATP to carbon 6 of glucose. Can you draw that nucleophilic attack? As I said, this is the one organic mechanism that will get you through my talks, and so it's absolutely in your interest to take a stab at this. You don't have to get it perfectly this time, but I'm moving on. Anytime you're looking at a nucleophilic attack, you look for two things. You look for a lone pair of electrons. They won't always be shown, but that'd be an oxygen, nitrogen, sulfur. And you look for a double bond. And we've already said which is which. We want the hydroxyl group of the glucose to nucleophilically attack the phosphorus in the double bond. Also crucial, the nucleophilic attack will happen to the central atom, usually a phosphorus or carbon. Oxygen will never be the subject of a nucleophilic attack, but it very often will be the attacking group. And so the lone pair of electrons on the hydroxyl group nucleophilically attacks the partially positively charged phosphorus atom in the double bond to oxygen, and those electrons resonate up to form the intermediate. So in that reaction, can you identify which of these is a nucleophile in a nucleophilic attack and which is an electrophile? Please pause and do so. Since the electrons are attacking the phosphorus, that makes the phosphorus an electrophile. The positive charge likes or loves the negative charge. The electrons are attacking the nucleus of the phosphorus atom, which makes the lone pair of electrons the nucleophile. Can you also then draw the arrows to show how this will resolve to kick off ADP and a phosphorylated form of glucose? Please draw those arrows. So every nucleophilic attack will have the same thing. There will be two arrows showing an attack and a double bond resonating up to an oxyanion to form the intermediate, and then you will sort of reverse those arrows to show the oxyanion kicking off a leaving group. Have you drawn it, please? I'm moving on. The lone pair of electrons in the oxyanion, and it could be any one of these three oxyanions, will resonate down to reform the double bond, and that will kick off the diphosphate. So in this case, the electrons will resonate from the bond between the phosphorus and the oxygen onto the oxygen to make an oxyanion, releasing the gamma phosphate to now be part of glucose, to make glucose 6-phosphate. Let's step back for just a minute and ask a question. The organic chemists say to me, wait a minute, why doesn't the hydroxyl group on the glucose attack this phosphate or this phosphate? And for that matter, there's 1, 2, 3, 4, 5 different hydroxyls. Any one of those could attack any phosphate. You should be able to get 15 different compounds out of this. And that's absolutely correct. If you have a beaker full of sugar and ATP, there would be some occurrence of a whole mixture of possible compounds. Please pause the video and tell me why all those different compounds, which are probably being formed right now in some of your cells, are irrelevant and don't matter at all. The answer is fairly simple. Random, uncatalyzed chemical reactions occur at an extremely slow rate. When you have these different compounds floating around in solution, the probability that any one of the hydroxyls will nucleophilically attack any one of the phosphates is actually kind of low. In contrast, the rate of a catalyzed reaction is profoundly faster, 1,000,000, billion, or trillion-fold faster. So for every trillion of the sugars that's phosphorylated on the correct oxygen, there might be one that's phosphorylated on the incorrect one. Furthermore, no matter how high the sugar is in there, the concentration of water is going to be profoundly higher. So what actually happens is what I told you about. You leave it sit for a couple hours. Most of you just get hydrolysis of the gamma phosphate and then of the beta phosphate and then ultimately even of the alpha phosphate. This stuff all hydrolyzes. It is the enzyme that grabs the glucose, grabs the ATP, and it specifically positions only the hydroxyl number 6 only adjacent to the phosphorus of the gamma phosphate. In other words, the specificity of enzyme catalyzed reactions are due to the very specific positioning. And you think about it, you need these electrons to be pushing up exactly at the right angle to be at the very bottom of the phosphate group to let the electrons resonate up here. So again, positioning is what prevents all these other random reactions from occurring. And if in your cell some of the glucose floating around happens to nucleophilically attack the alpha phosphate using, say, hydroxyl 2, well, your body would just ultimately recognize that as something unusual and would end up breaking it apart. But what we're really trying to form is glucose 6-phosphate. This is at the very beginning of sugar metabolism. And so this is an extremely important reaction in your body, and as a physician, you will need to be familiar with this. Now I should point out that hexokinase, of course, is trapping the glucose inside the cell. Glucose itself can enter and leave the cell through transporters freely. But once it's inside and phosphorylated, it can no longer go through those same transporters. This means that the glucose has gone up in energy because now it's a more complicated molecule with a negatively charged group. How did we get the energy for the glucose to go up in energy? Please stop and answer. And of course, that's the fundamental idea. If you have glucose and inorganic phosphate floating around, they will not combine together and form a more complicated molecule because that would be a decrease in entropy and it would actually be an increase in bond energy, so that it would require energy to form it. You can't just randomly form glucose 6-phosphate from a solution of inorganic phosphate and glucose. But in order for the glucose to go up in energy to glucose 6-phosphate, the ATP went down in energy to ADP. What does it mean, the laws of thermodynamics and going up and down in energy? Here, very simple. The ATP had a very high energy bond, the enzyme grabbed it and formed a slightly high energy bond with glucose 6-phosphate. The net reaction energy between the hydrolysis of ATP and the formation of glucose 6-phosphate, the net energy overall went down. It turns out that glucose, not the phosphorylated form, but glucose, is the most prevalent organic molecule on the planet. That is, in the polymeric form of glucose called cellulose, you have the redwood forests, an awful lot of the structure of the plants on Earth. And in the polymeric form of glucose called starch, we have half of all dietary energy for humans, so very biologically relevant. And when you break down glucose into the monomeric form and take it into a cell, it is hexokinase that donates the phosphate to the glucose that traps it inside the cell, the first step of metabolism. ATP, as I mentioned, is the central currency of cellular energy that you've heard a lot about. Not only is it the center of energy, it's one of the 4 nucleotides, and as we're going to talk about next, it is a precursor that's used as a component for many coenzymes. And in fact, to finish this talk off, will you identify the adenine base, the ribose sugar, and a phosphate group that shows that there's an adenylate group in each of the coenzymes shown on this page, which we'll be talking in about part 2 of coenzymes and vitamins. Thank you. As always, any questions, I really enjoy hearing from you, so don't hesitate to email me or call.